Now, for the next talk, I would like to welcome uh, Dr. Angelo Serani. He will be speaking on update on small molecules developed by the Italian Institute of Technology.
Thank you.
The floor is yours.
Thanks, Shiran. Thank you, all of you. I'm quite anxious when you have to talk to these beautiful people, but let me introduce the first result of this project that was founded by the Italian association, the CTNB1 Italia. Can you hear me? A little bit better? Okay, so I was saying, I'm very proud to be here and to show the first results of this project that we designed together between IIT, the Italian Institute of Technology, and Gazzellini Hospital, that is a children's hospital placed in Genova. And this project was founded by the CTNB1 Italia, the Italian Association of Family, and we are now just showing them also the results in advice in this moment. Well, let's start from the beginning. Our effort in the CTNB1 syndrome started a long time ago in 2014 when my colleague Walter Tucci discovered a mutation that was generating a mouse with a lot of syndrome, a lot of features that can be collected into syndromic features. That syndromic features that now we call CTNB1 syndrome. Well, in particular, the mutation was this one. It's quite hard here, right? Okay, let's see how to, anyway. So in the red mark, we have the mutation of the mice. And these mice, as you can see from this picture, generate a quite different neurons. As you can see in the wild type, you have this arborization, these highlighted axon and neurons that correctly develop. But in the mutated mice, it's a heterozygous mutation. We generate neurons with less arborization, less developed. And this feature led to brain structures abnormalities. As you can see on the left, the connection between brain regions are tight and not so well developed. And these abnormalities led to brain functions, cognitive function alterations. We have a bunch of differences from the wild type, since the child in the pups actually have reduced call towards the mother, so we have altered relationship between the mother and the pups. We have spatial memory and motor coordination alteration, but also contextual learning alterations. And if you take in mind yesterday, if I remember well, Nina described something like this feature can happen also in patients. So at that point, starting from that analysis, we found that at the beginning, the alteration in the brain connection was due to impaired connection between two proteins. One was mentioned also from Rafa yesterday, from beta-catenin and N-catenin and E-catenins, the catenins family in general. So starting from this data, we developed a project together with the CTNB1 Italia, and we designed a project that started from bioinformatical analysis that's take out some drugs to screen through molecular assessment in vitro in the cell, and then go out to be tested in our mouse model and human IPS, and the IPS derived from the patient that carrying the CTM mutation. So as I say, we started from the human interaction between beta-catenin and N-catenin. But at that time, we don't have the crystallization of the N-catenin, so we have to create models starting from analyzing the differences between the mouse and the human. We generated a minimalization thanks to Ida and Federico Falchi, our colleagues, and in this model we assessed the effect, the bioinformatical effect of interaction between catenin and catenin by adding molecules and looking what happens during the time in the tight interaction, and if whether was able to increase this kind of connections. So we started from 500,000 of drugs from a database. We go through this high throughput bioinformatics screening, and we found almost 1,000 molecules suitable for the wild-type beta-catenin, 1,000 molecules suitable for the several mutations that we produce in our model, and we started to analyze 28 drugs listed in here. We collected a lot of chemistry, biochemistry, pharmacodynamics, and pharmacokinetics predicted by the bioinformaticians, and we started to analyze in vitro. So we are looking for connection between two proteins. So we have to design an assay, a model that easily allow us to easily understand if we are able to increase the connection between beta-catenin and the N-catenin. So we created a model starting from three components. We have the N-catenin, the protein, the beta-catenin, and we have this venous GFP. GFP stands for the green fluorescent protein. This is a protein that emits green light when excited. So we cut the protein into half, and we decoded the DNA information. And then we joined half of the information of the venous to the N-catenin, and the other half to the beta-catenin. We included another fluorescent protein that is red. We packaged all the DNA information in one vector, the same vector that was explained just before me, and we gave the DNA information to the cells. So what happened in the cells? We have the red fluorescent protein that start to glow as red because it's a full protein, and we have the production. The cells take the information from the DNA and start to produce the N-catenin fused to half of the venous and the beta-catenin fused to the other half of the venous. And we have the glowing of the green only if we have the complementation of the two proteins, only if we have the interaction between the two proteins, because the two halves of the venous can be reconstructed and the venous can start to glow green, to glow green. And this is the phase that we have in the cells, the colors that we have in the cells. As you can see here in red, we have the cell, the full cell body, and the green body, the green spots happen just on the side of the cells, or at least if you see here, if I'm not able sure to point that, but you can see some green rounds. It means that we have the complementation of the BFC into the cell membrane. So why doing this? Maybe you remember this from yesterday that's shown from our Spanish colleagues. We are looking for two particular pathways. One is the interaction between the beta-catenin to the catenins, so we are talking about cell-cell interaction, and the other one is the ability of the beta-catenin to help other factors to increase transcription of genes, to activate pathways in the nucleus, and to allow the turning on of some information. Well, these two pathways are needed for cellular development, but we know that N-catenin in neurons is especially needed to the neurite outgrow the neuronal development. As you remember from the first slide, we have lacking of this neuron branching in our mice model. So in our opinion, by finding a drug that is able to increase the interaction between the beta-catenin and N-catenin, we can unbalance the two pathways in favor of neuronal growth and neuronal maturation. So what we did, we gave the DNA to the cells, we plated in the multiple cells, in the multiple plate, and we used an incucyte. Imagine the incucyte as a microscope built in an incubator. In this incubator, we can track from the seeding, from the plating and the treatment, along several days, several time points, the effect of the drugs on the fluorescence. And as you can see here from this example, you can see some red spots in some wells, so in some squares, and other squares remains shut down. So starting from these experiments, we collected our samples, we treated with all the 28 drugs that we found, and as you can see here, we have some drugs that are able to increase the complementation of the BIFC. So we have green lights normalized by other values, but we have still at least four working molecules. Well, this is an example of the molecules. We can predict some behaviors of the molecules starting from the structure, starting from these parts that are named residues. For instance, we know that these three fluoromethyl groups, the one on the left side, is very used in drug discovery to allow the molecules pass the membranes, so to pass the blood-brain barriers, to pass the cell membranes, and to reach the brain. But also we know, for instance, that this can be toxic for the liver. So in that case, we have a bunch of information from the molecules that now should be tested on our in vitro and in vivo model. Of course, we can reduce the concentration to reduce the toxicity. We can assess multiple administration, increase the incubation times, and we have to play a lot with these drugs to reach the final concentration suitable for going farther in mice and in the iPS cells. But we also tested another experiment. As you know, we have different mutations, and in particular, those signing in these pictures are represented the Italian cohort of patient mutations. And in most of the cases, the mutations of our child gain a stop, so you have a cutting of the protein. In some other cases, you have a frame shift, but it means that you gain a stop after 5, 10 amino acids. So we can assume that the frame shifts at a certain point behave like a stop. So what we modeled the interaction of the beta-catenin with the N-catenin by generating the mutation, and we found that at least we have space for interaction. The interaction is lacking because the real and the full strength interaction need the full protein, but at least we have some material to work with. We tested the drugs that we found effective on the first experiments on some mutation. We took a couple of mutations. One is the 333 amino acid. It means that this tryptophan stop codon is the blue arrow that cut the protein, the beta-catenin at half, more or less. The other one is another that preserved 2/3 of the protein. As you can see here, the bar number 7, it means that that drug is effective by to increase the complementation between catenin and N-catenin. So at this point, we are at the moment that we have to discover the off-target. We have to discover the side effect. So we prepare the samples and we pass the ball to Andrea and Lucilla from Gaslini Hospital. Well, they use a very nice procedure. Imagine that once a molecule binds the beta-catenin in this particular example, you need energy to detach them. So by increasing the temperature, by analyzing the peak of proteins, the weight of the proteins, you can assess the energy that you need to detach the drug we used. And this is help us to understand if we have the molecules bound to the beta-catenin, but also if we have the molecules bound to other off-targets in the cells. But this technique is very nice because in parallel, we also have the pictures of what happened in the cells. So each of these peaks represent a protein. So we know after the treatment, if we have increased pathways that we don't want to have. So we have a good pictures of what happened in the cells after the administration. This night I received an email from Lucilla and was very, very funny because we have a lot of data to analyze. And it seems that, okay, we are not touching stuff and pathways that are not so close to our target, but this needs further analysis and we have to look into the data in the deep. Well, once we assessed the the right molecules to use, we have our mouse models. We know the effect of the mutation on the brain, so we know where to look for. We will treat our mouse in different time points, in different ages, during pregnancy, after pregnancy. So we have a bunch of analysis to do on these mice to assess the real efficacy in a more complex model. And at the second, as probably Andrea showed you yesterday, we are able to produce a lots of brain organoids that we call mini brain that recapitulate some brain areas like cortex or like hypothalamus. In our laboratories, we gain this expertise to generate the brain organoids, but we also gain an expertise to test the organoids on a chip that is able to assess the electrical activity. In this cartoon, now that I hope it's moving, yeah, you can see how, this is an example from another experiment of ours, you can see how the brain reacts to a stimulus drugs. And from this information, we can see the connection between neurons in a more complex way in a brain that is made by human, in a mini brain that is made by human cells from patient cells. So it can be a good model to test the real efficacy of our drugs for future improvements. Well, this is our project. We designed this project in a sort of circular way. So at a certain point now, with the Gaslini proteomics analysis, we can define new off targets, but we can also start with new libraries. In fact, this is the first round, and we are ready because the another bench of drugs that we can be, in which we can start to analyze for the second round. But also, I want to use this occasion, this beautiful stage, and my colleagues like a call to arms, because the more samples that we have, the more cells that we have, the more mutation we can screen, the more effective we are in finding a treatment for a lot of different patients. So I'm finished. I want to leave you with two brief news from Italy. One is that we created the first advisory board for the CTNB1 Italia Association. The board will help the future of new founding and scientific advisory for the association. And the second one, I want to invite you on the next year in Genoa. We are organizing the second Italian meeting, and I hope to find you here the next year in Genoa. Thank you so much.